Good morning, everyone. My name is Eric. Today, I would like to talk about temperature, stress, causes host cell detachment in symbiotic Cnidarians, implications for coral bleaching. First, what are corals? Corals are Cnidarian animals in marine. They are marine invertebrates in the class as low as of phylum Cnidarians. They consist of two cell layers, an outer ectoderm and an inner endoderm. Ectoderm and endoderm, along with mesoderm, are the primary germ cell layers. Why are coral reefs important? They can provide a comfortable and safe home for many marine organisms. You can see this harmony picture. There are many fishes live in coral reefs. They support more species per unit area than any other marine environment. They are some of the most valuable and diverse ecosystems on Earth. But corals get into trouble. What problems are corals facing? You can see this photo. Corals in Great Barrier from 2012 to 2016. Coral bleaching in Great Barrier Reefs was even worse than expected. It is estimated 70% of shallow seawater corals has died because global climate changes. So what problems are corals facing? Seawater temperature has been rising gradually. Before we talk about how to conduct the experiment, let's talk about where does coral's color come from. Corals cannot make itself colorful. They need to dinoflagellate help. Symbiotic zooxanthellae gives corals colorful pigments. This is zooxanthellae. They are located in vacuoles within coral's endoderm. They can not only clean up the wastes, but also provide nutrients for corals. They are corals' good friends. But corals are sensitive. They would abandon zooxanthellae when seawater temperature changes. No matter seawater temperature is high or low, corals would release host cell to seawater. There are 5 different ways host cell detach from coral's endoderm, like exocytosis of host cell or bleaching of distal portion of host cell. Sometimes cell would naturally occur death or cell death because external factors. Finally, host cell detach from coral's endoderm. Okay, now we have an initial understanding about corals. Let's talk about our experiment. First, we should collect animal samples. Their scientific names are Aptesi potula and Pocillopola babiconis. We refer them as anemone A, and the coral P. Both of them are Cnidarian animals in marine. They live in 1 meter depth. Their habitat temperatures annually from 21 degrees Celsius to 27 degrees Celsius. This is Hawaii Institute of Marine Biology. Early experiments were being processed in here, and animal collected location is near here. Our objective, we want to know, first, mechanism of bleaching, second, morphology of cellular entities released, Third, why did coral lose protein by bleaching in excess of lead, accounted for by those Rosenzeller alone? This is our temperature treatment, and this is cold stress. We put our experiment samples in 12 degrees Celsius, and we put our control samples in 25 degrees Celsius. This is hot stress. We put our experiment samples in 32 degrees Celsius, and we put our control samples in 25 degrees Celsius. We want to investigate coral protein by bleaching, so we remove seawater from petri dishes. Then we do some chemical treatment, and we can get our experiment sample like this. This is anemone A, and our control sample is unstressed. And so was coral P. Our result, you can see this photo. In your left panel, hot cell released to seawater in response to cold stress by anemone A, and in your right panel, hot cell released to seawater in response to cold stress by coral P. Photo A and C is a hot cell released by coral P, and photo B and D is a hot cell released by anemone A. You can see these two photos. These two photos clearly show that hot cell plasma membranes like this, and the vacuolar membranes surrounding those cells, and there is hot cell nucleus and mitochondria. This photo shows that hot cell plasma membranes rupture. You can clearly see isolated those cells. This is the other photos. Our experiment, there is no photos shows that hot cell released after hot stress, because cellular entities released were too fragile to collect and handle, so there is no photo. Our protein determination results. In this picture, our x-axis is Rosenzeller release, and our y-axis is soluble protein release. And the black square is our experiment, and the control square and white square is our control. There is a significant positive correlation between Rosenzeller release and soluble protein release. In the anemone A, this is anemone A, and we can get same condition in the coral P. This line is positive correlation. Discussion. Our goals. First, mechanism of bleaching. Seawater temperature in short time is the primary mechanism of coral bleaching in our experiment. Second, morphology of cellular entities released. This photo I have shown in slides earlier. Third, why did corals lose protein by bleaching in excess of accounted by those and cells alone. We can detect that total soluble protein increasing after host cell rupture by temperature stress. Finally, take-home message. Corals are colorful because zooxanthellae gives them pigments. And when coral bleaches, it is not dead if we improve environment immediately. Thank you for listening.